So good morning, everybody, for coming this morning at the unusual time. So today is a pleasure, it's a real pleasure to have Carlos Meliá as a speaker. So, I mean, I'm just going to make a very short history of his different positions. So he did a PhD with Jordi Vascontes, but probably many of you at least know some of his contribution between the University of Alcalá and the Doñana Biological Station. Then he moved to the US to a center that I think it doesn't exist any longer, NCS, it's still
yes, it's still there. Yes.
So it's a National Center for Ecological Analysis and Synthesis. So he spent some time there, also with a fellowship from Microsoft, which is, I think, is quite unusual. And after his experience there in the US, he moved to Europe, to Switzerland, where he got a position at the ETH, specifically, it's called that. EABAC is the Swiss Federal Institute of Aquatic Sciences in Lucerne. And he also delivers some lectures at the University of Bern. And finally, he's with us because he was awarded a Maria de Zambrano Fellowship, what means that he's going to be with us for this year and the next year. And the idea is that he's going to help us to develop further the part of ecology, evolution, networks, and all this business. So I think if you want to talk with him, he's in the first floor.
Yes.
And that's it. So thanks, Carlos, for being reckless and accept the fellowship of the Maria Zambrano come from Switzerland to Spain. The floor is yours.
Yeah, thank you, Victor. Thank you, everyone, for being here. Also, people online. It's great. I would like to dedicate this talk to my son, Levin. I think he's around today. Maybe it's the first time he's going to watch a scientific talk. So welcome. Stop me anytime if you have questions, inquiries, critical feedback. So this is maybe a summary, a non-exhaustive summary of the last 10 years of my research about diversification. So it's essentially a teamwork. I have contributed a few things, but it's a much larger team. This started more or less 10 years ago at NCS. And the main reason was that at that stage, the neutral theory of biodiversity was starting to be, mainstream. And at the same time, there was a neutral theory of molecular evolution. And so we had many discussions in how to connect these two neutral theories to strengthen a neutral test, right? Not only for neutral molecular evolution or molecular evolution patterns, but also biodiversity patterns. So this is the motivation, and it's mostly a teamwork. So I'm going to be a bit bold today. So I have a new setting to give a talk. It's Jupyter Lab, and so this is the HTML, and I am testing a few things. So sorry if there is some error here, but it's nice. It's a prototyping talk. So I have five lines, five initially independent questions. What is a species? Is a fundamental question, not only in biology, but also in physics or in chemistry. Is why matter diversify and how it stabilizes at biological scales, but also chemical scales, physical scales. So it's a fundamental question. But if you take a look at the theory networks, diversification on networks is quite rare. So in most studies, just explore patterns in networks or dynamics of networks, but never diversifying, evolving with explicit evolutionary mechanisms, regardless the scientific field you are working with. So that's an important component in this talk. Then a classical problem in ecology and evolution is, does diversification increase in the tropics? Most people would say yes, because today the pattern is quite clear. I'm going to be adding some constructive criticism to this main pattern in ecology and evolution. Then why are there some lineages with many species while others have just a few? So we are transitioning here from macroevolutionary patterns to the microevolutionary patterns. So this is a big problem still, a big gap. So there are many lineages that differ in species regions, right? Many have just a few, and others, just a few lineages have many, many species. And why this is so, nobody knows, because if you account for geography, this happens in the same biogeographic regions. So there are other mechanisms entering in the diversifying lineages, in addition to external factors. And then we go to how do diversification and coexistence connect each other in complex ecosystems, see how to merge spatially explicit landscapes, network models to implicit evolution with evolutionary dynamics. And the last part is how does the next unifying diversification theory look like? It's a bit bold, this last question, but it's my view, my humble view about how things are going to go in the future. Just for those interested in extending, knowledge about diversification, for each question there are a few references that can help you to expand further a bit each of the questions. And this talk is online, it's a GitHub, so you can download and run and use all the slides. So for each question, there are a few references, and I think it's maybe important, right, to expand a bit my bias, right, about diversification. I'm going to tell you today. So what is a species? There are many definitions of a species, many. I'm going to use in this first question the most standard one. There are many transitions in population genetics, and today most people accept that the most robust definition is reproductive isolation. It's a cluster of individuals that are isolated from other clusters. Now the point is, which is the degree of isolation? Is 100% or is 10%? This is an open question. And it's an open question because many species originate by forming hybrids, by mixing genomes that have been diverging for a long time. And so this is quite interesting. The first problem, we then go to the question, what is a species? Is, okay, do species have a boundary? That's an open question. For example, this animation, you see time, x-axis, probability density of the phenotypes or the genotypes in red. So you start with the same point, stochastic processes, and you start to diverge. Just a mutation-driven process. And some genotypes, they go to the mean, they follow the mean of the distribution, the mean types, and then they drift, and they are rare. Then fitness enters in these dynamics. And depending on the fitness functions, you are going to be stabilizing, only getting the mean types or the extreme values when you have the advantage of rare types. This is the standard way, right? Most people think about species in biology, in ecology and evolution. Now, the point is how you define a boundary. Let me introduce you as one of the simplest toy models on graphs. I think graphs can be useful to understand the simplest, right, and the most robust definition of a species. So imagine you have a population of J individuals. So each row is an individual. And each row here has an infinite genome, can be quite large. And so you have many sites, and in each site you have a mutation rate. In principle, there is no linkage between sites. This is a bit more complex model, but at this stage, you have a population size, genome size, and mutation rate. And now you put these individuals in a graph, in a mating graph. And mating systems is one of the core problems in evolutionary biology, because mating systems determine many aspects of how a graph diversifies. And for example, here you have a cartoon with 8 individuals in red nodes, and the connection, right, is when the similarity Qij is larger than a threshold. You put a threshold. You can tell, well, but this threshold is arbitrary. Yes, it's arbitrary. You can get from data the genetic distance, right, at the population level between two populations and obtain this from the data, which is the threshold that it decreases dramatically the probability to have two individuals producing viable offsprings. So using this network approach, we can explore, right, what is a species. For example, we can have stochastic processes here, like demographic stochasticity, birth and deaths. And you can have many models, but this is the simplest one. Now you start to run, you start with, let's say, a population of fish or humans colonize a habitat, a few individuals. They are quite similar because they are at least a family, and now they start to diverge. And the divergence is just in sympatry, no spatial structure. It's just because individuals in the periphery prefer the neighborhood than the individuals in the other part of the graph. And then you start to have divergence, new phenotypes start to emerge. And you have more phenotypes. This is just birth and deaths, right? You are killing individuals with the same probability. You pick up one individual in a asexual evolutionary graph, or you pick up two individuals in a sexual evolutionary graph, and then you fill the empty space and you go along with generations. And with this process, you are essentially achieving speciation. You see clusters. And this is the most, let's say, extreme definition of a species. This graph contains 4 species, 4 completely isolated species. And
the reproduction is sexual, right? There's a kind of, and presumably the offspring has kind of a combination of the two.
Yes.
Don't you then, is that not a convergent effect?
You're adding, if this is exactly this, I think this inequality, these 2 potential solutions, exactly. So if given total population size, mutation rate, and the genetic similarity to have viable offspring, these 3 parameters, and if the third one is larger than the mean of the genetic similarity matrix, then you don't get clusters. So things converge and you don't get clusters, regardless the number of generations. Essentially what you have, this is the mean, which is 1 divided by 4 times total population size times mutation rate plus 1. Essentially, this is the mean genetic similarity value of the whole matrix. But if the Q mean is lower than the mean genetic similarity matrix, then you have the formation of clusters, because divergence is stronger than convergence. And at that stage, we can do an easy experiment here in an asexual graph chain. You have individuals that are reproducing, right? Produces one individual, you kill this individual, produces one individual, and with a given mutation rate, you can compute the number of steps that where this individual is a new species. You can technically do this, and you can do this for asexual populations and for sexual populations. In sexual, you are essentially adding a second randomly selected individual from this initial graph. And so now the point is that we can compute the speciation rate, or the number of steps in the asexual and in the sexual reproduction graphs, the values where you have a new cluster. And essentially, this is the number of steps, which is essentially when in asex, normally it takes more. It takes more if the combination, right, of the Q mean satisfy the cluster dynamics, then it takes more for asexual to reach a speciation event. And essentially 1 divided by the number of steps gives you the speciation rate. And we can prove that in the sexual graph, the speciation rate is higher than the asexual graph. This is the standard definition of a species in a graph using a toy model. There are many derivations and many potential additions that can strengthen or can indeed turn down this analytical expectation, especially if you add spatially explicit habitats, heterogeneity, or if you add linkage, right? You combine or you add crossover, then things become a bit more tricky. There are many people working in obtaining derivations, right, from more complex models, and see how they deviate from this simplest toy model. So we are here about what is a species, but the point now is that many people in evolutionary biology, in biogeography, ecology, they have been asking questions at the macro level, as in physics. That you have branches, right? Some people focus in the tiny things, other people focus in the macro things. And now, one of the most fascinating patterns in ecology today is the latitudinal biodiversity pattern. Yeah.
So this is a theoretical approach.
Yes.
But in reality, what do the people do? I mean, they are not trying just to define a species. They don't try and ask, does this animal is able to reproduce to the rest of the animals or not, right? Nobody does that. But when you try, you find a fossil, you don't do that. So how this definition, I mean, how do you know that this definition is good enough to capture what the species do? Or,
that's a great, it seems we discussed the talk, but we didn't. We didn't. So, but this question is the reason of this, right? Like second line of thought, right? There is a conflict. It's an explicit conflict because people working in macroevolution generally don't care about the microevolutionary details and the species concept at that level of definition that I was working in the previous section. So this is a gap. It's an empty gap now.
And the definition is also based on genetics.
Yes.
And not on traits, which could be additional definition. Right?
Exactly. Exactly. Yes. So traits, right? We can also define species at the trait level. So the point here is that they can reproduce, but they significantly differ because morphological traits or mating traits or interaction traits. You are going in this line maybe, but this is still to be done. So how, this is maybe the last part of the talk, is how to define a species in the next diversification theory, accounting for different levels. Yeah, that's a great, but this pattern is the pattern that we have today. So you have hotspots, red on the tropics, right? With some variation, but the pattern is quite clear. And then you move towards the poles, north or south, and you decrease the number of species. So the point is why? It's because speciation rate, diversification rate increase in the tropics because temperature, stability, plus other potential mechanisms, or is just a contemporary pattern that it is not related with deep time or, right, a general pattern in ecology and evolution. Indeed, it seems it's a temporary, it's just temporal pattern. Because if you go deep in time, for example, this is for the late Cretaceous dinosaur, latitudinal biodiversity gradient, there are also gradients like this for other taxa. You see here on the right side, in red, diversity doesn't peak on the tropics. And you can see also the landscape masses doesn't peak in the tropics. The landscape masses is more or less stable along the whole gradient. And so from paleo data, they have been reconstructing these curves and the pattern doesn't emerge. So the point is why? One of the options is the continental drift, right? Is one of the potential forces, driving forces that are externally driven diversification rates. Here we come to the external factors, right? It's nothing about the internal genomics, right? Or phenotypes or phenoms of populations. It's because external factors, you know, are essentially driven, driving the way the populations diverge. Is allopatric speciation. And we have been working in mechanistic models based only on habitat-driven diversification. But these models are a bit more complex because you have layers. You have a layer is habitat layer, and then you have the connectivity layer, and then you have a resolution of the data like a 10,000 years, 50,000 year beans, and you can run simulations accounting for presence, absence of populations in the sites that satisfy the habitat requirement, right? And the existence of a specific habitat during that period of time. And then you run simulation at a large scale, but genetics is missing. It's macroevolution. It's essential. You're interested in how the continental drift has been driving the evolution of hot and cold spots. And you can see here, in your left side is for corals. And you can see 40 million years ago, the green is the hot spot. There were many coral species in the Tethys Sea when it was open to the Indic Ocean. It was a much larger system full of diversification events or speciation events. The fossil data tells you that this was the case around 40 millions of years. And in the right side is the simulations, is the mean of many, many replicates, how the hotspots at that age emerge around the Tethys Sea and also the northeast of the Atlantic Ocean. And so then when you add this continental drift, you start to see that these seas start to close. Many species are essentially escaping, moving, and this is a question of currents. In our model, what the dynamics here is you have time steps changing the seascape, and then you colonize if you are a certain distance. There is a threshold. And speciation only happens if two sites have been isolated for a given period of time, and we fine-tune these parameters. And it's essentially allopatric speciation. It's driven by external factors. And essentially, what you get in the model and in the data is that the hotspot they have been moving towards northern Australia. And this is what you see the hotspots today. And you can track these processes. So this is kind of the second branch to understand what is a species and how diversification happens, but at macroevolutionary level. So the point is then that the latitudinal biodiversity gradient is gone in deep time for many taxa. And environmental gradients, they still can be an important force. Together with time and space, can be an explanation for diversification of most lineages. But why are there lineages with many species while others just have a few? Why? One example, there are many, many examples of lineages varying log scale variation in number of species. One example is the cichlids. The cichlids radiations is one of the lineages of cichlids have radiated in 25,000 years, like from 1 to 200 species. It's brutal, it's outstanding. But many cichlids didn't radiate in the same area. They contain only 1 or 2 species. So the question is why? Most people would say, yes, it's because lake size, because external factors. You can also apply this as a test. But there have been many tests, and none of them really explain well that external factors are driving the dynamics. So you need to account for internal dynamics. Internal dynamics here can be sexual selection, coloration, complex patterns in genomes like habitat selection, selection to resources, habitat specialization, and many other processes that, the microevolutionary theory, the beginning right of the talk went in that direction, come here in rescue to build a more compact theory, but it's not there. I'm going to present now a few models, not at that stage, that try to put these things together. So what if DNA, development, gene expression, behavior, and sexual selection, among others, combined with external factors to produce a complex set of interacting layers challenging speciation events? So we are there. And the question we can ask here is then, how do diversification and coexistence connect each other in complex ecosystems? One of the most, I would say, simplest and elegant experiments in experimental evolution is this one with bacterial communities. This is a long experiment, an old experiment. There are many additional experiments, like the Lenski, right, experimental bacterial evolution for many, many generations with many fascinating patterns. This was much shorter in time, but when you can see in the 3 plots on top is you see patchy clusters of bacteria. They have a patchy structure, and this is with similar resources. And then if you go to another level, you see morphologies. They have distinct morphologies. So they not only stand separated in a small petri dish, but they also, right, have different morphologies. So there is something around that. They are interchanging probably genes, right? They are using some kind of gene mixing to produce this convergence in the trait space. And then in the bottom, you have a 3D, and they also are in different positions in this 3D landscape. So there is habitat preference. how this heterogeneity originated is tricky because this experiment was closed. They didn't add resources externally, so it's a closed, right? It's a non-equilibrium. Sorry, it's an equilibrium experiment, right? They are not adding external resources to the dynamics, but it's an important experiment because you see that sympatric speciation, even in bacteria, can be possible, can be originated. So the point is that we extend the toy model introduced in the first section of what is a species in a spatially explicit landscapes, in a random geometric graph. Essentially, what you have now is a distance matrix. In red here, you have sites, and in each side, you have thousands of individuals. Initially, they are all the same, but now you have a spatially explicit landscape, and the landscapes differ because you have a random geometric graph, depending on the Dmax. Right? And depending on the threshold, you can have a fully connected graph, you can have a graph that is near to the transition threshold, or you can have an isolated, right? The infinite island model. All the sites are isolated. And so you then can run on these graphs at least 3 scenarios. One is the symmetric gene flow scenario. Individuals move symmetrically in the graph, right? They essentially, you randomly pick up a site, you randomly pick up individuals in that site, and then you move along if, right, you have connected sites to that site with the same probability. So individuals move without any history, no phylopatry, no habitat selection, no, it's a neutral diversification model. And so you can also add centripetal gene flow, which is essentially that you are moving from the periphery to the center. And you can have these 2 scenarios, right? You move, right, with some conditions, you move towards the center or you don't move, but you only move from the periphery to the center. And you can also move from the center to the periphery, the centrifugal gene flow model. So we run these 3 scenarios in these different graphs and see what happens. How many species are formed, and do we see hotspots, and which scenario do we see a hotspot? So this is the general pattern here. There is one motivation in this approach is connecting external, internal factors as a first step, but there is also a second element here. Is the classical island biography theory is one of the most successful theories in ecology. Essentially says that the diversification is in the periphery. So you have a global pool, it's a global area that is your rain and propagals coming to the islands, and the island you have the speciation events. But this approach here, we explore different scenarios. Essentially, we are going to see here quickly that when this is the distance to the center of the graph, you are in the center here, you are in the periphery here. It's an arbitrary matrix. And here the diversification rate, which is the number of speciation events minus the number of extinction events, because populations you originate but also go to extinction because just stochasticity, demographic stochasticity. So essentially what we have here is when you have the model where populations move from the periphery to the center, you have a hot spot in the center. Is this dotted line in red? And then as you move to the periphery, you decrease the diversification rate. The logic seems quite easy to understand, at least for me now, a posteriori. Essentially what is happening is that because you are bringing distant gene flow from many different populations along the landscape, all these populations are mixing the genes and are essentially producing a hotspot in the center. But when you are moving in the other direction, you are losing genetic diversity because you are not mixing with population in the other side of the graph. You are only just going in one direction and you are losing. Of course, you get a speciation, but it's much lower. You never get a hotspot. And when you have symmetric gene flow, you don't get a hotspot. Yeah.
Sorry, I just have one question. What is I got lost. What is the meaning of the center and the periphery in topology? So it's like the connectivity is
There is a pattern of a connectivity pattern in the center. Normally, the center for a given D max, for a given cut, that is going to determine the total connectivity of the graph. there is a correlation between being in the center and the number and the grid.
Okay, so this
In a 2D, sorry, in a 2D graph.
So here the center is a spatial location. So is the center of
You have like coordinates, exactly.
Okay.
You have coordinates.
And is linked with the topology of the
Exactly. And you can say that in a 2D, this area is the center and this is the periphery. No torus here, right? If you go to the one extreme, you don't come from the other. You just, it's like you move from the, let's say you go to the Canary Islands, right? You invade an island. You are invading an island. You are not really turning around the earth, right? And moving, you are just stuck there in the periphery. From the species range point of view, this is the underlying assumption here.
But this is like a meta-population.
It's a meta-population, exactly.
You have many individuals with different.
Exactly. Each site is a red spot, and each site contains, for example, in this case, 2 clusters. You see this one here, if you remove this link, if this individual dies, you have 2 species. If you apply the strong definition of what a species is, but if you relax, you can say that these 2 clusters are 2 species. But then you have a speciation happening, right? And you count the number of species emerging and the number of species also going to extinction. And these are the 3 scenarios, and this is the general pattern. So hotspots, essentially, given J, right, total population. This is total population of the meta population, the total number of individuals initial, which is keep constant during the simulations. Then genome size, mutation rate, the genetic similarity, right, threshold for mating, and from the probability to have a viable offspring, and then Dmax, what determines the connectivity between sites. And gene flow is essentially, right, coming later, right? If you have 2 sites connected, then with some probability you are moving individuals between sites. So with these dynamics, we have hotspots in the center and not in the periphery. We are almost done. Just a last, for me, one of the most fascinating problems today is the idea of convergence. For example, when I first saw this slide, I was completely ignorant and still. So these guys here and these guys here, you take a look and you say, hey, these and these are the same, more or less, phenotypically. So they are the same lineage, right? And the same with all the other species, but it is not like that. So this row, this column here, they belong to the same lineage. And this row here, they belong to the same lineage. The point is why? Is because externally, again, fact-driven factors, so they colonize different lakes, and habitat structure is driving these genome phenotypes to these states, and the system stabilizes. It's one hypothesis, but it's quite improbable because it's not easy to map the habitats, the dynamics of the habitats, and external factors. This can be also driven by internally, let's say, evolutionary graphs, right? Or genomes, right? A linkage or development, or the connection between genotype and phenotype maps, which is called essentially the constraints. You have developmental constraints. Essentially what this other hypothesis says is, regardless of the external forces, because you only have a few number of paths during development, you are going to produce only a subset of phenotypes, and they, in the same lineage, are going to convert to these states, regardless of the external dynamics. These are 2 hypotheses. There are more. But the point here is we have been working in coevolutionary diversification in mutualistic networks as a way to understand convergence. Here, now we don't have only one lineage. We have 2 lineages. We can have here like plants and pollinators. Let me explain briefly the model. It's individual based. You have here individuals, different phenotypes, different blue color tones. And now you have deaths, A, right? You kill individuals. Is again demographic stochasticity. B, you enter in sexual reproduction if you satisfy 2 conditions. If you are within a given distance in the graph, and second, if you satisfy the genetic similarity threshold. Then these individuals mate 2 plants or 2 pollinators. And then we go to C, which is a bit more sophisticated now. We have recombination. So now the genes are mixing. Now how you do this? There are many potential models. One easy model is that you split randomly and you mix, right, the DNA of the 2 individuals. These are all haploid individuals, not diploid individuals. but there are many potential and more complicated and realistic scenarios to model, right, this diversification process. But then we enter in mutation, and you have the offspring coming to the system. So you have a complete cycle, and you can then quantify the emergence of new genotypes and also phenotypes. The point here is that you have, right, two individuals, and you mix their genes, so it's recombination splicing, and then you have mutations, and in the end, the traits, you will add a little of noise, right, environmental noise to the system, which is essentially something to capture that fluctuations in environmental variables. So with this system, we can try to understand, for example, the convergence of hummingbirds pollinating a specific lineages of plants, right? The big size and the morphology associated with the interaction traits. And not only for the hummingbirds, but also for the plants, because you have the two lineages evolving. And essentially what we quantify was two matrices. The Q matrix is the genetic similarity matrix, which you have, you know, the genetic similarity between pair of species, and the phenotypic matrix, which you have also the phenotypic similarity between species. And you can compute these two matrices. And now convergence, for example, for the case of species A and C, you see they are quite similar from the genetic point of view, but quite distant from the phenotypic point of view. And you can have many scenarios. Essentially, evolutionary convergence is that you account for genetic similarity, right? You split in the tree, you put a threshold there, and then two species that diverged time ago converge in the phenotypic space. So they can be quite distant genetically, but quite similar phenotypically. And in this case, we can do this analysis with data. Right? We can do this for plants here. We explore a convergence threshold for the plants, for the hummingbirds, and then complementarity. Essentially here, the idea is that convergence, you quantify, right? When you are here to 1, you are quite strict. You have to have exactly the same phenotype value. We're using here only mean values. It would be great to have the distribution of phenotype values for all these species, but this is not available. It's almost impossible to get, but at least you can compare the means. And when you essentially explore this range, as you can see, the more relaxed you are in the threshold, the more similar, I mean, the prediction is within, right? The interval confidence predict, right, the observations for the plants and for the hummingbirds. And for the complementarity, we are also in a range of good predictions. This is all preliminary results, but the idea here is that you can extend, right, the dynamics, the molecular evolution in neutral landscapes as a neutral test for two interacting lineages and see if convergence and complementarity follow the empirical patterns. So, quick summary here. These are complex flowcharts, but I think this summary is amazing. Essentially, it says that you have time here along the rows, and on top you have space. And here in time, you have speciation stages, right? Here is absent, so two populations are the same. Then you start to have some divergence. Here is allopatric. Is the case of external factors. Two populations just don't have gene flow because you have a barrier or two sides have been diverging and populations cannot mix genes. And here you have non-allopathic. So populations do mix genes. And here you see that you have also the additional or different potential factors, like the standing genetic variation is how much variation the founder effect, a small population brings when it colonizes a site. And this, in addition to mutation rate, is adding variation, right, to these two scenarios. And you move along in time, you are gonna have more divergence in the allopathic or in the non-allopathic, and then you are adding new potential mechanisms, combinatorial mechanisms, which is essentially genomic processes. So because recombination and you start to have linkage in the graph point of view, you start to have genes associated That the idea here is that they are associated because they are selected, right, in specific habitats, and you have clusters of genes that are being selected. This is the idea of fitness in specific habitats, and because you have different habitats, you are increasing the speciation rate along this time trajectory. So this is a summary, quite simplified, but yet quite complex, of the existing conceptual framework of diversification, accounting for intrinsic, right, intrinsic genome-driven processes. I would say that the final summary here is more complex. And I think most of us do some modeling, some data analysis. We normally have a bias, some prefer toy models, or they're more complex models. It's an open problem. To me, the most difficult thing is to work with the two approaches and know well, right, the pros and cons of each approach. But it's a bit more tricky because when you discuss with people doing system biology, like gene expression, patterns are amazing. I mean, from the 70s, when the data came, people started to analyze humans and chimpanzee, and the genetic DNA was exactly the same for many regions. But then the gene expression was completely different. So it's evolution at two levels. And so even if you don't have a structural changes at the DNA level, at the expression level, you have changes that produce distinct phenotypes. And this is an important characteristic of genome evolution and also diversification. And so you need system biology, you need genomics, you need phenomes, which is essentially the architecture of traits that can be decoupled from genomes, because plasticity, behavioral traits, and many other features that are not imprinted at the genomic level. This is essentially called the idea of heritability. Some traits are highly heritable, others no. So it's highly heterogeneous. And the point here is that you also have external factors, but you need to put them all together. And this can be also be visualized like one row here is reproduction, mutation, and recombination. These are basic processes. I think I have been working at this level. It's quite frustrating because essentially we are in the simplest level. But the second level is when you add explicit architecture, because genomes and phenomes are complex. We have many traits. People say thousands, not only in humans. And so the point is we have a complex architecture that we don't know how it affects diversification. And this is an important level because it connects genotypes to phenotypes. And the next level is then the combinations. Probably most populations of any taxa contain many distinct genetic and trait architectures. And maybe this in the end is going to be important to predict why some lineages diversify quickly and others don't diversify. But we don't know. The last level here is selection. At which level operates selection? Here enters multi-level selection theory. It enters at the genetic level or at the phenotypic level or at the whole architecture level. Still open questions. And there are many gaps in our understanding of connecting the different levels to understand diversification. That's it. This is the take-home message. I try to summarize the different parts of the talk in three categories. Speciation is a complex process. Physical, chemical, and biological processes drive the emergence of new genotypes and phenotypes along diversification stages. The ideal would be to combine external and internal factors to produce heterogeneous diversification patterns in space and time. But we are not there. And these patterns combined with intensity and directionality of gene flow might play a critical role to anticipate radiation, how fast diversification occurs, and the formation of hot and cold spots. And finally, toy models, as I mentioned before, can be useful to understand basic speciation process, to understand the species concept. But current data suggests that systems biology, genomics, phenomes, fluctuating environment all together are needed to connect the micro, the internal dynamics of populations with the macro, the external dynamics, diversification processes, and biodiversity patterns. Thank you.
Thanks, Carlos, for the talk. Now we'll have some time before lunch for questions. So please.
Should I have...
Do people in the audience listen to you?
I think that they listen to me through the microphone. I think, no?
Say it again.
Yes, I think
They listen to you. I don't know. But you know, I repeat your question.
No, I think it's super.
Okay, super. Yes. Yeah.
So thank you very much for the talk. I had some questions regarding this metapopulation model that we were discussing before. Like it was inspired in this. You started with experiment, right?
Yes.
Yeah. So I have questions regarding the experiment. So I don't understand what was the initial condition. So you have a soup of nutrients and you put one species there and they diversify in like in genome and spatially, or how?
That's a great question. Here is the same resources in homogeneous. It's a petri dish, quite small, an initial clonal bacterial population, right? You know, accounting for standing genetic variation, which is right the diversity in the initial stage. They minimize, and then they release the population in an homogeneous environment, and they wait for a few generations. They contrast different waiting times. In some cases, this never was observed. So the stability of this experiment is not, you know, one-to-one. In many cases, they just most populations go to extinction. They don't stabilize. And for this pattern, they replicate many times, and in many scenarios, they found this pattern, but always keeping the same conditions. They didn't explore, for example, heterogeneous nutrients, like habitat-driven adaptation to resources or non-equilibrium systems. This is a bit more tricky. This is an equilibrium system, meaning that they start and then they close the system and they wait for a number of generations. That's it.
And so this is a spatial heterogeneity merging, let's say, from this uniform.
From the internal self-organizing somehow.
And these islands that we can see are different species, or
that's a great because bacteria interchange plasmids, and they have many complex, you know, processes underlying the genetic mixing. This is another experiment. I think it's late 80s, 90s. So, and this is maybe the standard experiment of sympatric speciation. You don't need isolated populations or heterogeneity to produce these groups, but the stability of these experiments is still under discussion. And the idea of a species in bacterial communities is complex because, I mean, in many studies, they have shown cluster formation, but the complete isolation, like in the model I explained in the beginning, with, you know, complete independent clusters, is extremely rare in bacteria. There is something around plasmids that they are able to mix even in extremely divergent. I mean, there are many experiments around this. It's quite an interesting problem, not only for diversification, but for the pathogen evolution, right? To bring in new toxins to the bacteria, right? And new defenses, right? In bacterial evolution, this is well known. This is almost an infinite problem because I have been reading only a few papers, not up to date here, but they mix extremely divergent bacteria with plasmids and they are functional. They recover functionality. So the idea of a species in bacterial is not fully, right? It's not a fully independent cluster, I would say.
Right. Yep. If I may ask just the last thing, in your model, how do you build your topology? Like is
here?
Yes. What? Yeah.
Yeah. Here
is that the dynamical network or just
it's a great
quenched thing? Or
here the landscape is static, meaning it's a 2D random geometric graph, and you tune the D max to produce these different topologies. From the fully connected, this is not an example fully connected, but it's a connected graph, and to completely isolated graph. And we run the dynamics. The dynamics I saw in the beginning, one graph, but in many sites. And so the dynamics in this scenario, you are having the dynamics in isolated sites. It's the same model than in the beginning, but in each site, you have, depending on the initial parameter combination, you can have clusters or not, but no migration. But in here, you can have migration because you have clusters of networks. in the space. And so you have migration depending on the migration value, right? Depending on M, the gene flow, we explore here a sub-range of parameter space. The complexity here is a toy model, right? It's a 6-parameter model, I would say. In biology, people mind is a toy model, a 6-parameter model. Maybe in the context of physics, it's already a very complex model, but we didn't explore the full range.
Thank you very much.
Yeah, you're welcome.
So, more questions? No? So I have one. So when I listen to this kind of talks, I'm a little bit confused at the end. Because, well, first, I don't know if the first model you introduce is useful or not. I mean, in the sense the one is able to reproduce from the data, and what the model is not able to reproduce from the data, right? So I'm thinking, for example, that, well, the reproduction is not, I mean, I think it's not linear with the genetic similarity, right? I'm thinking in brothers or sisters, right? The reproduction there, I think you have a payoff. So you also have a Q minimum to be able to reproduce or to be satisfactory in reproduction, right?
Yes.
So I don't know if this is important or not for the, and also I think I understood that the problem on the diversity of the gradient was moved to the gradient of habitats. Somehow, or this is what I understood, that the problem of the gradient of diversity is not so, but it's due to the fact that you have habitats, and the habitats are patterns that move in geological scales, and then it is a consequence of that. So I don't know if my understanding is correct, but in that case, the question would be why the habitats are distributed, or how the habitats are distributed across the longitudinal gradient, and why they are only those gradients and those habitats are no others, right? So what I want to say is that, well, I mean, there are many questions with respect to the approach between the theoretical modeling and the reality.
Yes.
And that I have many more questions now than at the beginning, right? So this is just an example. So I don't know if, how to navigate there.
So let me be as brief as possible, right? I think the first question, can I erase here? I think I can, right? Quickly, maybe the other side, because here I'm close to this and it's gonna be, right? Like I don't know how this is called, resonance, when I'm close to the, yes, so it's gonna be, but just quickly, I think Victor's first question relates to inbreeding depression, right? So essentially you have the Q, right? The Q mean can be from 0 to 1. We are saying here that, right? Only these individuals, right? Individuals that are, right? This is the threshold that are in this range can produce viable offspring. If you diverge along, right? If you move from this similarity, then you cannot, you can mate, but you don't produce viable offspring. Now the point is, what is the evidence for this? We explored many models. This is a strong inbreeding depression because the more similar you are essentially mixing more and more similar individuals. And we know this is a problem in many populations, right? Inbreeding depression start to produce individuals with a lot of problems. There are many cases around this, not only in humans. And so the alternative is to use a distribution here, right? Something like this. So you essentially have a range, right? From where you, in probabilistic terms, you produce fertile offspring with a given probability till a peak. And then if you are beyond, right, a threshold, right, depending on the variance, then you don't produce fertile offspring because you die. This is an additional, right, a scenario, but this contains one parameter more at least. So it's a bit more complex model, but we explored this, and this is a whole discussion, right? But this, we didn't know how to do this analytically. We can do this by numerical simulations, and the results, they are qualitatively similar, but we didn't explore many shapes, right? Or the strength, right? The decay, this decay, the strength of inbreeding depression, is what is the probability that the offspring that the offspring die. Right? And there is a lot of data on this. I mean, experimental data, for example, in fruit flies. In fruit flies, there are many data. It's the same plot. They have been, accounting for genetic diversity. They made many pairs, and then what they observe is essentially a threshold. When you diverge beyond a threshold, the probability to have viable offspring decays quite fast. These are for sympatric populations, sympatrically diverging populations of fruit flies, and allopatrically diverging populations. The threshold is in the two scenarios. So there is something with the evolution of genetic incompatibilities. And we enter here in classic models in population genetics from the 40s, 50s, that were essentially trying to understand why the evolution of incompatibilities. Humans, many pairs don't produce fertile offspring. It's well known. And it's mostly because many factors, external stress, many variables, but also internally incompatible molecular structures. And this is an interesting problem. But so we explore different, this is the first question, sorry, I'm maybe taking, and the second, I didn't get much your question, but I agree with you, it's confusing because essentially it's the open problem between connecting microevolutionary processes where you have well-defined assumptions and variables, and you can do some maths, versus macroevolutionary problems. Maybe it's the opposite that in physics, correct me if I'm wrong here. When you go to the macroevolutionary patterns in biodiversity, nobody has been trying to develop models, meaning equation-driven or general equations-driven models, stochastic, deterministic, to produce global patterns of biodiversity. It seems trivial, but maybe this is the reason of the confusion, right? Maybe the deal here is, okay, let's try to put theoreticians and empiricists, and let's try to connect microevolutionary models on graphs, stochastic demography, and environmental stochasticity to the general diversification models in broad spatially explicit landscapes. This is not there. Maybe this is the reason of your confusion, but if you work from the quantitative point of view, this is an open problem. Yeah.
Go ahead. Last chance for the last question. No.
One.
You showed some results on asexual reproduction, but then on the discussion is on the possibility of having offspring. But so it seems to me that it is more of the concept of genetic dissimilarity, so that you can have viable offspring to asexual reproduction. But you have asexual reproduction, that makes no sense. No. I mean, you can have a distance, but the idea of having viable offspring is lost for asexual reproduction.
Yeah, that's a great question. So your point here is that because this amazing capacity of bacterial or asexual reproduction systems in absorbing almost any gene pool, then this doesn't make sense. This is your vision here, right?
Yeah,
somehow.
That the discussion went to and you define or assume a reproductive isolation, and then you have some results on asexual reproduction.
Yes.
And asexual reproduction doesn't have this, no? I mean, but I think it's
yes, I agree.
It's actually the question that you were posing, no? That,
yes.
Well, we stick to this that we can say about sexual reproduction, but then extend it to asexual. But so there's a need for something else more fundamental for being a species.
Yes, maybe this relates with Tobias' question in the beginning about the convergence divergence. So for any taxa, if the internal diverging mechanisms, right, exceed, right, the externally or internally converging mechanisms, then regardless sexual, right, but this is the whole dynamics, and this is the reason that the stochastic processes is like birth and deaths. You can have some intuition around how these processes are happening in asexual. It's simpler, right? And analytically, they are much simpler. I mean, in most networks, correct me if I'm wrong here, you deal with asexual populations, right? You just have, you have like a new nodes coming from randomly selected nodes with some variation here and there. even if there is no explicit maybe evolutionary processes. But with sexual, it's a bit more tricky because you need to take account two, or you can even triplets. This is where more complex. I mean, I know some approaches now in networks working with triplets or quadruplets, and why sex is around 2 and not 3 or 4 or 10. That's an interesting problem, right? Maybe initially this was common in networks. You needed the pool of many individuals to have offspring, but then because many processes, this was selected against till 2 or 1. It's a minimalist. It's the extreme of the but this is and there are many taxa where you have a copulation of many individuals still, and it's not completely close that you only have, right, in the end only 2 individuals, right? So it's an interesting problem for evolutionary biology on graphs. So I think this doesn't respond completely to your question, but the asexual is the limiting case, the simplest, where you, even if not realistically, biologically, but it makes sense theoretically to compare with when.
What's the definition of a species in that case? You say species are defined by can reproduce together.
Yes.
Two individuals, if it's asexual, that's not definition.
Well,
why is that equally different from some other? Why is that one species?
That's a great question. It's about the gradient. One extreme is when you have complete isolated clusters, but maybe they are not there. But they are not there, but they are not in the other extreme. Because now let's try to develop a model, and we bring gene pools from many different pools, and some are functional and others not. This is the point, because you are not in the extremes. You are in a graph, which is something in between, but we don't know where. E. coli is able to absorb many variants, but at some degree, you don't. It's not functional. So there is a limit. And there are some experiments around this. So you are not in the boundaries. So your point is, yes, what is the point here of a species as an isolated cluster? Maybe it doesn't make sense, but it makes sense in a gradient. Now, derive analytically the gradient given the data. This is open. You see my point? You get a distribution, say, well, most species are between 0.3 or 0.4, something like that. This is, we don't know. That's a good question.
Good. So,
yeah.
Thanks for attending and thanks for the talk.
Yeah.
Have a nice afternoon.
Thank you, everyone. Yes.
Que en los controles no se escondía.
No lo sé cómo hacerlo. Lo que comenté del prototyping.
Sí, sí. No,
we go for
el resto súper bien.
Yes. At 2. Or what is? Yes, but what is okay for you? At 2 or even because we can also. It's close to the Simona's one. You know where it is, and I can also come to you because we go for lunch, and we arrive and then we meet. You are here, right? You are in the last one, yes.